You, Sean. Good evening, everybody, and thank you for joining us. My name's Dr. James Evans, and on behalf of our Brain Tumor Division in the Jefferson Multidisciplinary Brain Tumor Center, I'd just like to welcome everyone to the first of 5 weekly webinars that we'll be hosting during Brain Tumor Awareness Month. Brain Tumor Awareness Month is intended to recognize the vast number of patients that are afflicted with brain tumors, their families and friends that support them, and the medical personnel that dedicate their lives to the fight against brain tumors. Tonight's webinar is Glioblastoma, an update on current clinical trials, and it's hosted by my great colleagues, Dr. David Andrews, Dr. Kevin Judy, and Dr. Wenyin Shi. And so with that, I'd like to turn things over and look forward to tonight's webinar. Thank you.
Hey, Jim. Can everyone hear me? I assume so. So David Andrews, I'm a neuro-oncologic neurosurgeon. I've been at Jefferson for many years, actually, established the division in 1995. Dr. Evans was actually the second part of that division, and we've grown considerably since then. Sean, can you drive these slides? So I will try and be brief, but I must say I'm trying to unpack the immune system and relate that to glioblastoma. So I don't want to overwhelm. I think what I'm going to do is just some of these slides to really have an interest in going into the weeds. We probably have copies of this so we can share with you later. Next slide. So this is always just of interest. How the immune system works, and it really starts with physical barriers that our skin and our mucous membranes that line our digestive or respiratory reproductive tracts, which is which is the 2-foot focus. That's our front line from reproductive individuals that are trying to get into our bodies. Any individual that breaches those physical barriers is created by an innate immune system. This is the second line of defense. It's innate is a defense that all animals just seem to have and have evolved over 500 million years. Given that long evolution, the system of cells has a razor-sharp ability to recognize danger molecules from an invading bacteria or virus, or actually anything that's not self, and we'll get into that. And then if that's not enough, there's a third level of defense called the adaptive immune system. Next slide. So the adaptive immune system takes over where the innate immune system can't go any further. And there are teams of cells that work in a very complementary fashion, depending on what the environment is that they're seeing. But there are 2 essential types of cells that probably are involved in glioblastoma. So these cells are actually cells that actually create antibodies that attack extracellular pathogens such as bloodborne viruses or bacteria. T cells will recognize and destroy contaminated cells with viruses replicating within them. So outside of cells, think B cells. Inside of cells, think of T cells, both of which actually will work together to actually eliminate an invader. And we've been talking about outside invaders. Next slide. So cancer immune system. So this is the inside system. You're sitting on the report again. So this is a way where a normal cell sort of becomes a turncoat and becomes a cancerous cell. And I'm not going to go through this slide in great detail, but just suffice it to say that with enough mutations in a particular cell, that cell sort of escapes the normal program for how long a cell lives and the cell becomes not only immortalized, but it actually divides very quickly, and you get a sort of a threshold number of cells, and you actually have a malignant tumor. Now, the most important thing that we're going to talk about is what is it about these tumors that allow them to escape the immune system, because they're no longer serving the host. They're no longer. If you have a brain tumor, it's like, where's the immune system to take away something that doesn't belong there? And they're a very complex question, but it's probably down to very simple. The cells that we think are most important are the T cells. So they're the cells that will actually destroy a particular cell based on its contents. But T cells won't recognize tumor cells because, again, the surface of the malignant tumor cell is still red as self, so the T cell will not bother it. There are examples where T cells will recognize a tumor, so it's in other words, it's not self. But in that situation, there's another step that the tumor cell would have to actually be capable of interacting with the T cell. So the T cell actually becomes exhausted and falls off. So a cytolytic T cell response, or T cells that will kill the cell, are just not existent, and these T cells become exhausted and the tumor escapes the immune system. Next slide. So there has been progress, and Jim Allison in Houston got the Nobel Prize for the CTLA, which is the cytolytic T lymphocyte antigen 4. And this actually changed many cancers, but very few in solid tumors. So if we look at cancer, we think in terms of solid tumors, which are solid masses, or liquid tumors, which are, cancers in the bloodstream that are involving the cells, typically a B-cell lymphoma. The majority of cancers are solid tumors. They're much more common than actually liquid cancers. And malignant glioma is actually a very small subset of them. But malignant glioma is a sudden transition horrible disease that actually, when the Cancer Genome Atlas was formed in 2000, and we actually sequenced the entire human genome, this was the first tumor that was actually sequenced. And actually, that's a subsequent slide that we've been mining those data for now 25 years to see what we can make of that clinically, and how we can take our understanding of our of our DNA, and how that could translate into something that has actually clinical value. So regarding gray matters and the promise for a breakthrough in malignant glioma, success for immunotherapy, only a subset of these mutations that gliomas have in many solid tumors, but now we're going to talk about gliomas. So it's not that it's a mutation, it has to be a translated mutation. So going back to biology 101, DNA is transcribed in certain regions. They're called introns and exons, but exons are the portions of our DNA which are then actually transcribed into messenger RNA. And messenger RNA increases the protein, which is the end product of that gene. So mutations, if they're mutations that are similar to the normal sequence of a protein, they have no impact on the immune system. The immune system thinks that's self. But if the mutation changes the structure of the protein that's actually translated, that is what the immune system is actually going to respond to. Next slide. So synonymous is same or self. I think non-synonymous is different or non-self. And again, that's a slide for you to actually ponder if you would like a copy of this lecture. Next slide. So going back to the Cancer Genome Atlas, this was actually one of our colleagues, Dr. Castle. His colleague is my chief scientific officer at the company that I formed with Jefferson. And this is just what are called the antigen inclusive. These are the proteins that are changed by the mutations. And 98% of these mutations for each tumor in each patient is unique to that individual. So when you think in terms of targeting a particular protein, let's now focus on glioblastoma. The likelihood of having success by targeting just one aberrant mutation is actually going to be slim to none in terms of having an overall response, because there are many other cells in this very heterogeneous population of cells in a glioblastoma that are not carrying that targeted mutation. So the ideal solution is to take a glioblastoma in this case and actually take the entire antigen payload of that particular patient's tumor and treat that patient. Next slide. Just click that. We excise the tumor. Tumor cells are then treated actually in a facility that we've developed next, very close to Jefferson. I'll pause there for a moment. Remember how we talked about how cells have a normal program in their lifespan. There's a surface receptor called insulin-like growth factor receptor 1, which when activated will prolong the life of the cell. And cancer cells, including glioblastoma, overexpress this surface antigen. We develop not only we take these tumor cells, we dissociate them into individual cells, and then we treat them with something that attacks the surface receptor that stresses those cells, those cancer cells. They don't necessarily die immediately, but they're now stressed. Next, keep going. It's animated, so before the next slide, you'll see some. So we incubate those cells with this molecule, which is knocking down the insulin-like growth factor type 1 receptor. We put these cells into a small biodiffusion chamber, you see upright. It's a little smaller than a dime in diameter, but if you stack up 3 dimes, that's about the thickness of it. And it has a very small exclusion limit. So the FDA did not want us to take living glioblastoma cells from the brain and put them in the abdomen. Even though glioblastoma cells really don't thrive outside the central nervous system, the FDA they don't take that into consideration. They do not want cancer spreading from one location to another based on what you do. So these cells are then encapsulated in these very small chambers, and the chambers will actually jail the cells. Water can fuse through these chambers through a small biodiffusion membrane, but that membrane is 100 nanometers, which is well below the size of a tumor cell. So only very small molecules can get out of that chamber. So we actually jail the cells in this biodiffusion chamber. We radiate the chamber because the FDA wanted another really comfort level. So we would radiate the cells so that if we move these cells from the brain to the abdomen, the cells ultimately won't survive and won't be a threat to the patient in the surgery. So 48 hours of surgery, we actually make incisions in the abdomen and we implant these chambers. The chambers remain in the patient for 48 hours, and then we explant the chambers, because what happens in that 48-hour period is the tumor cells are driven into what is called an immunogenic cell death. So it's a regulated cell death where all of those translated proteins, those non-synonymous proteins or their peptides, diffuse out of the chamber and into this immediate surrounding area or the wounds that we create in the abdomen. So now let's just go back to our immunology 101. So we've now breached the physical barrier of skin. So now we actually open the abdomen, we place a foreign body between the sheath that covers the muscle of the abdomen and the muscle itself. And that immediately piques the interest of the innate immune system. There's something going on. Out of the chamber, actually all of the immunogenic molecules that this tumor has, in addition to the antisense molecule, which is also in there. And that gets the immune system, the innate immune system, really geared up. And that's where this whole process starts. So we train the innate immune system to respond in the direction that we want it to, which is to actually initiate a response of killing the tumor in the brain. After 48 hours and all of these, the full payload of antigens in these peptides have now interacted with cells called antigen-presenting cells, which are sort of the principal players in the innate immune system, that then traffic out of the field and go to the nearest lymph nodes, which in this case are the lymph nodes in the groin. If we explant the chambers after 48 hours, there's not a living cell left. So the whole process takes place within 48 hours of an initial hospitalization for treatment of this disease. Next. I think the final slide is the chambers are explanted. The patients then go for 6 weeks and then get adjuvant treatment, which is radiation and chemotherapy. Now, we designed these incisions below the belly button because it turns out the belly button is sort of it's a lymphatic watershed, which means our lymph nodes are gathering all the extracellular fluids that don't go back through the veins to the heart. They actually go to the lymphatic system, to the spleen, and other organs that process these cells, the secondary organs being the lymph nodes. So if we implant these chambers that are now allowing the diffusion of all these very antigenic proteins south of the umbilicus, they're going to be going to the lymph nodes, which are actually pristine. If you look at the brain and the fact that these tumors, even though they're supposedly hiding behind what's called the blood-brain barrier, they're actually not. They're creating blood vessels. They're very porous. So there are, in fact, we call it antigenic dandruff, which is whatever's coming out of that tumor is going to into the lymph nodes and periphery in a gradient. So we want this process to take place in an interaction between the innate immune system and the adaptive immune system. The lymph nodes are carrying all the T cells that are going to respond to the antigen that they've been presented with from the innate immune system. Next slide. I'll quiz you in the last 3 minutes. So this is just a recapitulation of what I've just described, and this will lead into the phase 1b trial that we published 3 years ago. Next slide. So this is the summary of that. Just to make it very simple, this was phase 1b trial that was based on safety, but it was also an escalation of the product that you're trying to use to treat the tumor. And you can all imagine that typically it's a drug, and the drug is escalated in dose. These are called phase 1 trials with the dose escalation, where you're looking for a dose-limiting toxicity, which ultimately you hope you don't see until before or well after you've already got a good clinical response. In this case, the drug is what's coming out of these biodiffusion chambers, which is undefined. It's this full array of antigens that are unique to each patient. So randomized patients based on the number of chambers implanted and the amount of time they were implanted, as you can see here. In the bottom right cell, you see that there were a lot more patients with 20 chambers in 48 hours. That was because there was no difference in safety when we did an analysis of this between these 4 cohorts. But the 20-chamber per 48-hour group were doing much better clinically, and as I'll show you radiographically. Next slide. So this is the summary. ITT stands for the intention to treat population, and to the far right is standard of care as has been practiced now for just about 20 years. So we included patients that would never have been included in a prospective randomized trial based on this standard of care. So we treated patients older than 70, and I think 70 is still young. But due to miss the far right, which is the standard of care as practiced then and still now, we wanted to be, well, we were actually beyond that, but in the phase 2 trial, we stuck with 70 just because we wanted to compare apples to apples, and that's the phase 2 trial I'll discuss in a moment. So there wasn't much difference in the intention to treat population if you look at overall survival, 17.3 months versus 16.2. But if you look at median progression-free survival, there is a significant separation. Now, if we begin to look just at the highest dose cohort, we're now seeing a separation in overall survival, 25.3 months versus 16.2 months, and we've trebled progression-free survival. So progression-free survival is the amount of time to initial treatment and the point at which the tumor starts growing again. Overall survival is just that. You're either doing well or you have not survived this disease. If you go one more to the right, and we look at only what are considered the STUP-eligible cohort, which is the standard of care as practiced, we've now doubled median overall survival and maintained progression-free survival. So we weren't expecting these clinical responses. This is corroborated by the MRI scans, which you see in the next slide. On the left are gross cell resections, and on the right are subtotal resections. So the red circle describes the area of enhancement of one of these tumors, glioblastoma. It doesn't mean that the whole tumor is within that red circle, but we as neurosurgeons consider growth hormones removing that enhancing element because it has impact on overall prognosis. So you see in the postoperative scan in the second column that you don't see any further enhancement, but that doesn't mean there's no tumor, residual tumor. That's why we have to proceed with adjuvant treatment or radiation and chemotherapy to actually treat a broader segment of that region, a broader radius of that region, because they're tumor cells that are migrating beyond the initial resection. And what's quite amazing that I had never seen before is at much later scans, there was no recurrence of these sites. So median time to progression in the literature is about 9 months, and for these patients, it was not yet reached. Subtotal resection, which is the panel to the right, was of greater interest to me because I could see residual disease and how residual disease responded to this treatment, which again is a treatment that takes place in the first standard of care hospitalization with craniotomy, but we're now introducing this treatment within that, in a 48-hour period in that hospitalization. There is no treatment after that other than the standard of care that we must adhere to. So you can see in the post-op scans, the top row, it looks like that all of that white spot is gone, but in different slices, there's still tumor, so it's subtotal resection. But what was interesting here was that we saw with residual tumor, a very slow regression of the tumor over a very long period of time. So if we look at subtotal resection in another publication, the time between surgery and intervention to progression was only about 6 months. Or intervention after the adjuvant treatment, I should say, was about 6 months. For the patients that actually, in these patients with residual disease, it was really almost 33 months. And this was, we believe, an immune response. So if you were to read the paper, we have a lot of exploratory objectives to support the fact that this is an immune response. Next slide. And this is the phase 2 trial. Again, adapted from the highest dose cohort, which is 20 chambers for 48 hours. Next slide. This is the clinical trial. I'm not going to drain this slide because the last slide you'll take a picture of, which is how to access this trial on clinicaltrials.gov or one of the advocate groups. We have many advocate groups that are very much involved in this trial. Next slide. For the phase 2 trial, I'm happy to report that really we're almost within 14 the goal of 100 patients, and these are the institutions that have participated. Next slide. So our passion is patient care, and that's why we're here tonight in front of you. This is Brain Tumor Awareness Month, and I couldn't be more honored to be in front of you to talk about this. Next slide. That's the summary of what we've talked about, and again, we'll make this accessible to those of you that really want to dig into it further. I think the next to last slide is just our contact. Next. All right, so thank you for your attention. I don't want to take Wenyin's time. Jim, go ahead.
Thank you, David. Great talk on the new development of immunotherapy in GBM, particularly with Jefferson's contribution to this very exciting study. I will share my slide first. And I think thank you everyone again for sparing your evening to attend this webinar series, which is the first of our month-long weekly series for the Brain Tumor Awareness Month. So I'm going to switch topic a little bit, talk about another endeavor that Jefferson has heavily involved with, and this is we call the fourth modality for the GBM treatment, tumor treating fields. As you know, conventional treatment for GBM involving surgery, radiation treatment, and a systemic therapy, including immunotherapy. But now we actually have another effective option to treat the patient with a very novel, completely out-of-box approach called a tumor treating field. So when we talk about GBM, as everyone is aware, it is very aggressive primary brain cancer. Even though the incidence is not very high, it's only about 14,000 in a year in US, but this is disease carry a very high rate of recurrence and progression and threat people's life. Despite the endeavors of all the care providers in researchers in the field, the actual advances of treating this disease is actually quite slow and limited. And you can see, if you look back from the past 50 years or so, and we have stepwise increase of survival, mainly based on the combination therapy. Simply adding radiation treatment for surgery, we almost double the survival rate of our GBM patient. Then in the early 2000s, we introduced a new chemotherapy agent, which is obviously the standard care now, temozolomide. By adding temozolomide to radiation treatment, we actually increased the median overall survival for another 2 more months. Now, most recently, the development is using the novel treatment modality called a tumor treating fields. By adding the tumor treating fields to the maintenance temozolomide after patient finishing chemoradiation treatment, we actually increase overall survival by about another 5 more months. So what is tumor treating fields? Tumor treating fields is a novel physical wearable device treatment. As you can see, this is an example of patient wearing this device. The patient unfortunately need to shave their hair to attach those arrays onto the scalp. And those arrays will be attached to a battery pack, and it will produce a alternating electric field that actually impact the cancer cells. So how does it work? For the physics characteristics of that, it's actually quite relevant to radiation. It's all part of the modality called electromagnetic wave. But it's actually electromagnetic wave actually can be a lot of different things, actually dramatically different physics and biological effects, including radio wave, microwave, infrared, our visible light, ultraviolet, X-ray, gamma ray, and also the tumor treating electric fields. The difference is it is a very long wavelength, And so this is an example of how different from radiation treatment. So radiation treatment has a very, very high frequency. The wavelength of that is super, super short. But the electric treatment field, the wavelength is about 1,500 meters. So it's not something that we are able to focus on tumor, but the field effects that cover the brain that lead to the benefits. The mechanism of action, the first and well-established mechanism of action is actually impact the tumor division or the mitosis. So when those pulsating electric fields that's flipping back and forth in the brain, it will affect all the molecules that carry a charge. So in this particular settings, it's actually affecting the DNAs. So when the cell undergo mitosis, there will be a spindle structure forming to pull the DNA apart and lead to the cell division. But when the electric field is impact on those molecules, the spindle structure cannot form properly. As a result, tumor cells cannot go through the division properly and lead to the tumor death. Interesting enough, it was the more investigation we have for this devices or the treatment modality, we gain more knowledge about this treatments. Actually, now we know it is actually has multi-modality mechanism of actions. As mentioned, the most important mechanism of action is anti-mitosis, basically stop the tumor cell from division and lead to cell death. But it actually has a lot of other very important mechanism of action, including it will interfere the cell permeabilities, and it will inhibit cell migration. It can inhibit DNA damage repair and induce autophagy. And actually, most recently, we also know tumor treating fields can interact and enhance immune response. And so this treatment device is actually started into the treatment for GBM in the early 2000s. Initially, it was approved for patients with recurrent GBM, but the most exciting breakthrough is really coming through the trial focusing on patients with newly diagnosed GBM. So this is the pivotal trial of EF-14, which is a 700-patient phase 3 randomized trial. In this trial, patient will be randomized to have radiation treatment with temozolomide followed by temozolomide treatment versus radiation treatment temozolomide followed by temozolomide plus the tumor treating fields. And as this, you can see the survival curve of this trial. You can see the survival curve actually separating from the very beginning and remain to be separated. And as a result, there's about 4.8 months overall survival improvement for the patient who received the tumor treating fields treatment. More excitingly, we also seeing that the tumor treating fields are more than double the patient who was able to survive more than 5 years. So we are also getting more patients into the long-term survival group. Unlike the chemotherapy, it often has certain molecular markers can predict responsibility. It's interesting enough, this tumor treating fields will work very well, equally well, despite different patient characteristic and molecular characteristics. For example, for the patient who has MGMT methylation or non-methylated promoter, patient who has different extent resection, or a patient who has different age group or different gender, they all respond equally well, means they have a similar range of response, about 30% improvement overall survival. And because it's a wearable device, as you can imagine, the more the patient is able to use it, the more treatment they have. So that's actually reflected in this result as the secondary analysis of the study as well. We actually can see if the patient who has increased compliance, it means they are able to use the device more royally, the more compliant they are, the better the outcome. If patient who is about 50% compliant, we are able to see an improvement compared to patient does not use the device. But the more the patient able to use it, the better the outcome. If we see if you look at a patient who are using the device about 90 to 100% of the time, and the survival benefit is almost is more than double. And so this dramatic improvements of the outcome. And so how do we want to take this treatment into the next level? And this is, what excites us when this new modality become available. We brought this treatment to our patient, offer this to our patients, but we also would want to take this to a next level. So interestingly, some of the basic science research showing that tumor treating fields can inhibit tumor DNA damage repair. Which is very exciting because we know radiation works through DNA damage. If the tumor treating fields can be used to inhibit tumor DNA damage repair, basically it can potentially use as a radiosensitizer, or it can be used to make radiation treatment more effective. And this is first proven in the cell culture studies. As you can see, those patients can have tumor treating fields alone or radiation treatment alone, but when they combine those two treatments, we see dramatic improvements compared to monotherapy. And there's also a very important temporal relationship, means the DNA repair can happen very, very fast after radiation treatment, and you need to start the inhibition very timely. So when we give the tumor treating fields right after radiation treatment, we can see even more benefits compared to start tumor treating fields about 24 hours afterwards. So if we give radiation treatment and give tumor treating fields right afterwards, we potentially can achieve a synergistic benefit. So based on that, our group set up and research to introducing the tumor treating fields with concurrent use of radiation treatment. The first thing we actually did is, using our collaboration with our physics group, look at the feasibility and the physics impact of using tumor treating fields while patients have radiation treatment. And this is the first study published that in the world by our group, showing that we can safely give radiation treatment through the tumor treatment field array. Basically, the patient can have tumor treating field treatment and have radiation treatment, and they do not have to take the array off while have radiation treatment, and radiation treatment can still be delivered very, very effectively. And after this initial physics feasibility study, we quickly set forward to run our phase 1 trial, and this is the first in the world as well. It's a phase 1 single-arm trial run in Jefferson, and we enrolled 30 patients of newly diagnosed GBM, and this is the general overall schema. As you can see, patient who has newly diagnosed GBM, they will start the conventional radiation treatment with 6 degree and 2 gray fractions, and they will receive the temozolomide, but they will start using the tumor treating fields, which is by the commercial name Optune, at the beginning of radiation treatment and continue on for up to 2 years. And we also developed quite a few innovations for this particular combination therapy. First of all, we have had done physics study showing that we are able to deliver radiation treatment through those arrays, but in order to have that, core combination to be executed in clinic, we have to do some, modification for the patient setup, and we developed this particular setup system to accommodate the array. Basically, we put a foam pad underneath the mass which stabilize patient's head, and those array can be fitted underneath, and the mass will still fits well. And another innovation our group developed is scalp sparing radiation treatment. The reason when we actually implement this is the most common side effect for the tumor treating fields is actually scalp irritation, which is anticipated. People wearing those bandit-like arrays on their hair 24/7 continuously for months, and there will be some skin irritation. So about 50% or more of the patient will develop some degree of skin irritation. Radiation treatment can certainly lead to skin irritation. Radiation treatment will lead to hair loss and scalp irritation. So they can potentially be overlapping toxicity. So in order to achieve that, to mitigate those potential overlapping toxicity, we come up with a special radiation treatment technique called scalp sparing. Basically, we are able to delineate the scalp for the patient and use it as a special avoiding structure. So using advanced radiation technology, we can deliver radiation treatment dose to the tumor, but still minimize radiation treatment dose to the scalp. And based on our study, we actually can reduce the scalp dose and more than 50% by this special technology. And this paper is also published. So this is our feasibility data. Basically what we find out is this combination is quite tolerable. And even though we see a relatively high percentage patient will have some scalp irritation, but those scalp irritation are pretty mild, means that it is usually grade 1, and it will not interrupt the radiation treatment delivery, and often does not interrupt with the tumor treating fields delivery either, just need to have some skin care. And this is our publication, which is first in the world with 30 patients. Right after we published our experience with 30 patients, Israel group also published a small study with 10 patients with similar approach. And that data is actually very similar than what we have. Means we actually seeing a majority of patients will have some low grade of scalp irritation, but majority of the scalp irritation are in a grade 1 or grade 2 range. So it's not interrupting radiation treatment, and the patient can successfully tolerating the concurrent treatment. So based on that, there's a very large international phase 3 trial, which was carried out. It's called Trident, a phase 3 trial with EF32. This trial included 950 patients and is run in North America, Europe, as well as Asia. And in this trial, the experimental arm is exactly what we did in our phase 1 trial. So they adopt everything we did in the phase 1 trial and use this as a experimental arm, and run it an international trial. And our group still stay actively involved in this trial development and execution. And also the trial was open at Jefferson. Despite the trial actually activated right at the time of the pandemic, the trial is actually enrolled on track. And at the beginning of this year, this trial actually successfully enrolled 950 patients worldwide. And obviously, we are very, very eager to hear the result of the trial, hopefully within a year and a half to 2 years from now. And it can potentially further improve the treatment efficacy of combining tumor treating fields with radiation treatment and chemotherapy. So what do we take from there? So we have our phase 1 study establish a new treatment combination modality, and from the phase 1, we have to successfully take this to a pharma-sponsored phase 2/3 trial and finish and accrue this trial. So what's next? As mentioned earlier, tumor treating fields has been found to interact with immune system to induce anti-tumor immune response. So basically, it can improve or induce immune response against the GBM. So the mechanism of action is actually due to some of the things I mentioned earlier. The tumor treating fields can potentially interfere the permeability of the tumor cells. And in the normal cells, the DNAs are in the nucleus, which is trapped or wrapped within the nucleus membrane. But when the patient is receiving tumor treating fields, the membrane get disrupted. So there are DNA structures leaked out from the cytoplasm. When the DNA leaked out of the cytoplasm, it can induce a specific anti-tumor response. And obviously, if we have an anti-cancer specific response, we can potentially amplify that. And we have a lot of options to amplify that. The most well-known will be the immunomodulated agents, such as the drugs that are widely used in lung cancer or melanoma, the checkpoint inhibitor. People may be familiar with those medications, pembrolizumab was one of them. So that's exactly the idea when the Florida group put forward this single-arm phase 2 trial with about 26 patients. So in this setting, the patient will still receive the standard care treatment, means radiation treatment and a chemotherapy with temozolomide. And then they will go on to the tumor treating fields with maintenance temozolomide. But besides that, they also introduced the immuno checkpoint inhibitor, which is the pembrolizumab. And based on this phase 2 trial, we can see the patient has dramatically improved progression-free survival. The progression-free survival for this group is about 12 months. The historical control from the EF14, which is the largest phase 3 trial to establish the role of Tumor Treating Field, the PFS is only 5.8 months. The overall survival also dramatically increased from 14.7 months, the benchmark, to the 24.8 months. So this is a very exciting single-arm trial. So based on that, now we are actually rolling out the new phase 3 trial, EF41. This is a phase 3 randomized trial that including placebo control use of the pembrolizumab with the concurrent temozolomide and the Tumor Treating Fields. So this trial is sponsored by the Novocure, and Jefferson will be one of the first sites to open this trial, and we anticipate we're able to open this trial in the end of this year. And this is also a very large, almost 600 patients phase 3 trial. So the patient will go through the standard care treatment with radiation treatment, chemotherapy, then tumor treating fields with TMZ, but they'll be randomized to either receive the immunocheckpoint inhibitor pembrolizumab versus a placebo. And we're very excited to and anxiously waiting for this trial to open in the next couple months. All right, I'll wrap up here, and I'll just save a couple minutes for question and answer. Thank you.
Gentlemen, we do have a couple questions. First one came in during Dr. Andrews' presentation. It said, is this for newly diagnosed glio? Define newly.
I'm sorry, this is newly diagnosed glioblastoma.
And what would you define as newly?
No interventions. The first intervention is always craniotomy. So we will accept patients that have had a needle biopsy, but not an open flap.
Great, thank you very much.
Sure.
One of the questions is, excuse me, it seems most clinical trials are for first-time glio patients. What is available for the second or third-time recurring patients?
I mean, either Wenyin or I could answer that question. All of the trials are for recurrence because there's no standard of care roadmap. The upfront trials are much less frequent. Actually, Wenyin actually framed the Trident trial, and I've framed the trial that we've developed as well.
So that's exactly Dr. Andrews mentioned. Jefferson, we're focusing on two special cohort patient. One is patient who have newly diagnosed GBM, means they just have a diagnosis with surgery, but have not received any other treatments. And we have a variety trial open for them, including we recently closed EF32, the ongoing INVAX trial. We also have a scalp-sparing trial looking at study to treatment to mitigate potential toxicity. And then the other group of patients who we active looking for trial for those patients was actually when the patient, unfortunately, after they go through the frontline treatment, develop recurrence. And there's several trials we actually evaluated. The current one we have open here is one trial through NRG Oncology Group, the BN010 trial. This is trial combining hypofractionated focused radiation treatment with an immunotherapy agent. They're introducing two different type of immunocheckpoint inhibitor and combine this with radiation treatment. And certainly there are other trials we are currently evaluating and previously opened here. including we actually did a phase 1 trial looking at combining reirradiation with different type of agents, such as HDAC inhibitors, aurora kinase inhibitors. So we are constantly evaluating those trials and try to bring this to our patient.
Thank you. We have a few more questions. Would you expect IR-AES in the EF41 trial? How do you plan to mitigate?
So the immunotherapy treatment has been widely adopted. So as mentioned, the most successful story is in the melanoma, and they actually using combination treatment in pembro and nivo and ipi-nivo. So and also they've been widely used in other cancer setting, including lung cancer. And so the experience of managing immune-induced toxicity is actually very mature. In this EF41 trial, we only introducing one immuno checkpoint inhibitor, pembro alone. So the toxicity profile will be much better compared patient get ipi-nivo with dual checkpoint inhibitor. And there's also the phase 2 trial, which has been run for an efficacy, early efficacy study, but also have a very nice toxicity profile study. So I think overall, the monotherapy with the use of mono checkpoint inhibitor with pembro in the setting of GBM is actually very well tolerated. And also our neuro-oncologists and other physicians are very well prepared and equipped to managing the immuno-induced toxicity.
Thank you. Another one we have in the queue is, if you are enrolled in a trial already, do you qualify for the TT fields later on?
So clinical trial is highly regulated, and so there are specific inclusion/exclusion criteria. So each trial has its own rules, and when patient in the trial, we have to follow all the rules. And so they may or may not allow tumor treating fields. For example, the INVEX trial does exclude patient to receive tumor treating fields, and in other trial, for example, the NRG trial was upfront GBM trial looking at chemo dose intensification. In the experimental arm, they will not allow the TT fields, but in the control arm, they do allow the tumor treating fields. One of the concerns for those newer innovation trial is there's a lack of information regarding how well the patient will tolerate if we're introducing a new agent while patient receive tumor treating fields. So the simple answer is variable, and it's really a trial by trial basis. And if you're on a trial, we have to adhere to the trial specification.
Thank you. Next question, has stem cell therapy been considered or used in glioblastoma patients? If so, how?
David, would you want to take this?
Yeah, no. So stem cell therapy is not, it's not really gained traction at this point. So stem cell therapy involves taking cells from the patient, expanding them, getting them back into the patient. It's a paradigm in which there has to be, if nothing else, differentiation and propagation of cells outside the patient and then bringing those stem cells back. But if you start expanding cells from a particular patient, every time if they grow and there's a population doubling and it's called splitting the cells into new, to expand them, it may have a different phenotype. So as far as the FDA is concerned, if you put cells in culture from a patient, every time you grow those cells in vitro, the next population of cells could be a different actual treatment. And that becomes actually insurmountable in terms of FDA regulation of what your end product is. It's an undefined product. So if you're dealing with autologous cells, you expanding a particular culture, particularly for stem cells, at least in the realm of glioblastoma, is really challenging.
is all the questions we have currently.
Currently,
If anybody has any additional questions, please add them to the chat or the Q&A. We have a few minutes left. Thank you.
John, if I could just make a postscript statement. Apparently, the audio did not work very well. I don't know if many of our participants didn't hear all of this.
There were some audio issues during your presentation. Did the best we could from my end to clear up the audio, but it seemed to clear up towards the end of your presentation, going into Dr. Shi's presentation. I don't see any additional questions. Gentlemen, if you have any closing comments, feel free. We have a minute or so left. If not, I look forward to talking to everybody next week at our next seminar or next webinar, sorry.
Yeah, I just want to thank all the audience again and to participate in the webinar. And brain tumor is a tumor has low incidence, but certainly has a lot of challenges. And our group here is dedicated to the care of the brain tumor patients. And thank you for the support.
I would second what Wenyin said. Thank you.
All right. Thank you, everyone.
Thank you, everyone. Have a great Wednesday night, and we will talk to. Oh, looks like we just had another question come in. Don't know if Dr. Andrews, Dr. Shi, you still here?
Are you still here? Yes.
Can a frozen tumor sample be used in IMAX approach?
In the index approach. Not yet. We're working on cell production, and you can freeze and thaw cells, but we have to go through a preliminary process of getting fresh tissue from new glioblastoma patients not on trial. It's called our fresh tissue protocol, which is how we actually set the foundation for what we're doing in this phase 2B trial. But I think the important point, and it's a good one, which is if you can preserve cells, freeze them and thaw them, and they're still viable. And I think they can. I'm absolutely sure they will be. But when you're talking about CGMP production and you're using those cells at a later date, we have to prove to the FDA that in fact it meets all the safety requirements and all the other criteria that they will look at to approve that. But I think that's a really important question because certainly tumors recur, and in fact, the recurrent tumor can have a different array of mutations. It could be a different tumor. It's histologically the same, but it could be at a molecular level different. So if you did a re-resection, we as neurosurgeons will only operate on patients that will benefit from a re-resection. So that's the first issue. But if we do re-resect them and we have this capability of finding out what that tumor phenotype looks like, the cells that we froze, we can just then decide whether or not they would be useful or not in a retreatment. But that's what we call life cycle management of a new protocol and a new treatment. So that's on the road.